Thank you so much, Amy, for that great setup for what will undoubtedly be a very interesting session. My name is Mia Levy. I'm at Vanderbilt University Medical Center, and I'm going to talk about the AACR Project Genie Consortium as a member of the Vanderbilt member of the steering committee of the AACR Genie project and the chair of the operations committee for that program. All right. These are my disclosures, some research funding, some consulting. There you go. I know I have to give it enough time. This is the new requirement. Have you read all these papers we don't give enough time for people to actually read it when we fly by? Okay, there we go. So, so what is the AACR Genie program? The AACR Genie program is an international pan-cancer registry built through data sharing. It is driven by openness, transparency, and inclusion, the goal of which is to improve clinical decision-making by linking clinical genotypes to clinical outcomes. We started with 8 founding members, which has now grown to 19 members that span North America and Europe, with plans for future expansion. The program is sponsored by the AACR, the American Association for Cancer Research, both internally and with collaborative projects, as well as grants and philanthropy, and a little bit of federal funding. These are the current 19 participants of the Genie Consortium. All of these have been groups that have what we call the Coalition of the Willing, groups that have self-organized with AACR's help to become members of a consortium under the premise that we are all going to share data that will then become freely available to the public and help drive clinical questions that we wouldn't otherwise be able to do by ourselves. So I think, Amy's, one of your last points was that big data then becomes really, really small data. Is what is motivating a lot of us to participate in this is that while each of our own institutions may have thousands of patients that we've sequenced to date, when you go to ask a particular question, the numbers get vanishingly small. So there's a motivation for groups to say, my institution alone can't answer this question. If we pool together, maybe we'll be able to do that. So that's part of what has motivated all of these groups to come to the table. The AACR Genie project was conceptualized by Charles Sawyer when he was AACR president in 2014. And over a course of about a year and a half, managed to convince the board of directors of the AACR to put seed funding together to fund this program. And the initial 8 groups were invited to participate, again, the Coalition of the Willing. And by November of 2015, that we had our first public announcement of the program, and about 13 months later, we had our first public release of data. That is actually remarkably fast when you really think about it. Getting all of these groups to come together, data use agreements, master participation agreements, legal documents, governance structures, and we were all doing it on a voluntary basis. So really an amazing feat when you really think about it. So Genie will be 3 years old later next week. And since our first release in January of 2017 of 18,000 cases, we're now on our 4th release, which was just this summer, with 48,000 samples released. And that was just from our first 8 member institutions. We've now added another 11 institutions and anticipate that our next release this coming January will be over 60,000 cases. So pretty exciting progress to date. As I mentioned, governance has been an essential component of trying to figure out how to get these groups to work together. So the program is sponsored by the AACR Board of Directors, and we have an external advisory board as well. And then there's a steering committee with all of the members of the Genie Consortium who make decisions by majority vote. We have the AACR acts as the coordinating center and has hired staff to be able to run the program for us. And we have several strategic partners, including 2 technology platforms, CBioPortal out of Memorial Sloan Kettering, as well as Sage Bio Networks that does significant work in collaborations and collaboratories. And then we have just structurally several subcommittees, including the Data Use and Publication Committee, a separate subgroup on pediatric oncology, which you'll see why in a second we need it, a participation committee, as well as an operations committee. So what is included in the Genie Consortium today as far as what are we actually doing? So I mentioned that we were able to put all this data together in a year, right? And publicly release it. That's because it's a very specific data set, okay? It includes the somatic tumor DNA of 47,000 tumors from 8 centers and a very, very, very small amount of phenotypic information, including the type of tumor like breast or lung, the histologic subtype when appropriate, like small cell versus non-small cell, basic patient demographics, and vital status of the patient. So really the clinical data or the phenotypic data is very, very small and very thin, which allowed us to get together quickly and put it out. But it is the most expensive data to collect, ironically, right? Because the Genie Consortium is actually not doing the sequencing as part of its data model, as part of its participation model. This is sequencing that's already been done as part of routine clinical care in a CLIA lab. And so that data is ironic. We all thought it was going to be the most intimidating data when it first came out. It turns out to be the most digital data on the planet, right? So other than maybe imaging today, those 2 are really great opportunities for transferring data. They have good standards for which to share it. So we leverage the fact that all of these institutions had the availability of their genomic data in a digital format that they could share. But the phenotypic data is actually a lot harder to get. The clinical data is a lot harder to get. So we didn't want perfect to be the enemy of getting something accomplished. We wanted to be able to put out some data that we could at least begin to show the process. So, and the data is made publicly available 12 months after the sequencing date. So the rule of the project is that, and I'll show this in a second, the sequencing comes out, and 12 months later, that data is going to be in the public domain in a de-identified HIPAA limited PHI way. The second half of the project is that we have sponsored research projects where we do go very, very deep, and we have specific questions that we want to ask as a group, where we take those 47,000 cases and we distill it down to the 150 that are going to allow us to ask a specific clinical research question. And then we do a deep dive into the clinical phenotype, and we fund the institutions to do the clinical annotation of however many cases they have at their institution. So so the question will drive how many patients are in a particular cohort. And then that data also becomes, once it's published, becomes part of the public repository as well. So I mentioned, so our workflow process is that each of the institutions will deposit their data in 6-month cycles. And then Synapse puts all that data together and does a data normalization effort for us. And we have a common ontology that we're mapping for diagnoses, and then the genes are normalized as well. And all of the PHI is removed, so we have a very limited PHI data set. And then the institution gets to keep their data to themselves for the first 6 months. And then the consortium has a consortium version of the data, which we get to use for the next 6 months to answer questions, just to give us some additional motivation for membership, right? And then the data becomes publicly available. Again, 12 months after the sequencing date. And it's currently publicly available through the CBioPortal, which I'll show a screenshot of in a second, as well as up on the Genomic Data Commons of the NCI. So this is a screenshot of the CBioPortal. So anyone can go to the Genie website. You need to register an account and promise that you're not going to try to re-identify patients, all these provisions that we've taken. And you can browse the genomic information that we currently have available, and I'll show a couple more slides on what our content is in a few minutes. And you can also download the data directly from us if you sign the data use agreements and the limitations on it. So the second half of the project is really these specific studies. So we then do these clinical queries based on a specific question that wants to be asked. And we go out to the consortium members and we create a contract and we say, will you please annotate the data from these 25 patients in your institution and 120 at your institution? And we come up with a common data model for that. We're using REDCap as our database. And then we do the analysis and report it back to whoever the sponsor of the project is and eventually make that publicly available as well. So as you imagine, the true value is going to be, however, once we can really get deep clinical data on a lot larger number of patients. And so this is currently just the patient journey that you showed is a perfect example of it. This is just another visual representation of the same kind of concept. But being able to get treatment information and outcomes information is really where we're moving towards right now to be able to do this for a much larger population of our patients. We have something called the Biopharma Consortium with the goal of doing manual curation of 10,000 of our Genie Consortium cases in the next 2 years. And that is going to require us to come up with a consistent way of recording outcomes and treatments such that we can train the curators at various institutions to be consistent in how they're doing it. It's not an insignificant task. So the Genie of tomorrow, we hope, will have not only somatic alterations, but perhaps germline DNA. It brings up a whole nother set of challenges with respect to public release of information. Circulating tumor DNA is not currently included. We hope to include that as well. RNA-seq data, epigenetic data, all of these are sort of coming into the clinic in a more standardized way. We expect that wave to continue to come in. And then, as we mentioned, the phenotypic data coming in. So we really anticipate that in a very short period of time, within the next couple of years, we will have 100,000 cases. And this really gives us the opportunity to do data for discovery. And we're hoping that the 10,000 cases that we manually annotate will also be useful for the machine learning community by virtue of being able to link the annotated data back to the raw information from the pathology reports, radiology reports, the response assessment, clinical note, and be able to bring that back and enable that data to become available to machine learning so that with the goal of no longer, I would say, machine augmented curation as opposed to 100% manual curation. So I just wanted to highlight some of the challenges within the program. This is just an understanding of the fact that each of our institutions has a different panel, or maybe we have some overlap in our panels, but not complete overlap. So much so that there's only 8 genes that are covered by all of our institutions. And this was only our first 8 institutions evaluated in this analysis. So you can understand that, but there's a lot of overlap of the other genes, but it's amazing to think what people think are important. Now, this might be because there's some smaller panels that might be focused on solid tumors versus other smaller panels that are focused on hematologic malignancies, and they don't actually have a lot of overlap, and that's reasonable and clinically appropriate. But just being able to normalize this data when you're doing panels that are so different is one of the strengths and potentially one of the weaknesses of the Genie program compared to something like Foundation Medicine or Tempest that have standardized platforms on all of their patients. They have the same platform or a variation of their platform as it migrates. But this is the real world, right? If you're trying to use real world data, this is the diversity that you're going to get in the genomics even. This gives you an understanding of where the data is coming from and the largest players at our Memorial Sloan Kettering. You can see the high percentage from there, as well as Dana-Farber is the second one. And the rest of us make up just like a small amount of it. But we all appreciate the contribution that we make because even if MSK tried to do it by themselves, they still need the additional 12 or 15 cases that we can offer in order to make a compelling outcome. I loved the conversations that were happening about diversity and gender. We have a very small percentage of cases that are African American across the entire data set to date, as well as Asian. Very, very small compared to the white population. And then we have a whole bunch that we have no information on because the Europeans could not give us that information. Their requirements were that race could not be shared as part of their data contribution. Another interesting societal challenge when you're doing these types of projects. And then this just gives you a sense of, if we just look at the orange dot, this is the Black population, and you can see which institutions have more or less fraction of their patients in that population. So it really depends on where you are and whether or not you have a patient population to support diversity. It becomes a main issue that these data sets aren't enriched for those populations at this point. Age is another challenge with us. So we have, this is mostly an adult population. And again, institutions give us everything they have, right? So they are not saying, I'm going to eliminate my pediatric cases by any means, but sequencing for the most part has been done in the adult population for the most part, which is why our average age is 60 years. But we do have, quite a few pediatric cases of under 18, but we're looking for collaborations that will enable us to enrich for that patient population in particular. These slides are very similar to some of the other ones you saw earlier out of California. The distribution of samples by cancer type is exactly mirroring which diseases have clinical indications for doing sequencing in the first place. So it's not too surprising that non-small cell lung cancer is the largest, followed by breast cancer, which is mostly done for clinical trial purposes as opposed to standard of care purposes, which hopefully with that PIK3CA inhibitor that someone was saying is soon to change. But this is our current distribution, and you'll see that we've broken it down by whether or not the patient's primary tumor was sequenced, a lymph node metastasis was sequenced, or a distant metastasis was sequenced. TP53, as everyone has already shown, is the highest, most frequently mutated gene in all of our data set by quite a lot, followed by KRAS and the other usual suspects. We are able to do sub-analyses, though, by disease population, and we've done several of these. This is just an example out of the non-small cell lung cancer population. And again, you can see, what's sort of interesting about this is, why is this whole thing gray? That's because these genes weren't tested at those institutions, right? So the common genes, fall to the top that are tested across most of the population. We've also done a landscape analysis of clinical actionability within the Genie Consortium patient population, showing that in the green is level 1, meaning standard of care therapy. So these are GIST population with close to 60% having a KIT mutation for that GIST population. Followed by melanoma with BRAF mutations with clinical actionability. And then the other levels would represent standard of care in a different cancer. Those are the blue ones, as well as promising therapies in other disease types. We also looked at the clinical trial potential actionability of our Genie Consortium population and found that for over 600 clinical trials that were enrolling for targeted therapies, that in our initial Genie population of 18,000 patients, over 16,000 had some potential of genomic actionability from a clinical trial that was currently available. We applied that then to the NCI match clinical trials that were available at the time. And this was a very interesting study in clinical trial feasibility, looking that these are all of the arms of the NCI match that were open at the time we did this analysis. And our patients would have matched just by their diagnosis and their genomics to most of the arms in the trial, except for these bottom ones, which you see out of 18,000 cases, this one had 0, this one had 10, this one had 18. Those arms are probably not going to accrue patients. And to be able to use a data set like this to do even just basic feasibility assessments for clinical trial eligibility shows that, perhaps there's some arms of a study that just aren't worth opening because you're just not going to have enough patients to accrue. Amy articulated many of these challenges of using real-world data already, but we've found them in our own experience as well. There's a lot of missing information, especially when you're limited by the EMR of a single institution, and these are major referral centers, and patients are coming and going, and we miss what happened to them once they've left, or the data, once they came before us, was in scanned files in the electronic health record. There's missing information of the other biomarkers, not just the genomic data, but there's a lot of immunohistochemistry biomarkers that are harder to find in the record. There's no consistent way of recording response to clinical therapy or the reason why treatment was discontinued. Lots of things in the electronic health record are recorded as fuzzy dates. When you have a breast cancer patient who's recurred after 10 years of being in a disease-free state, you say they got chemotherapy in 2005. The official dates no longer are particularly relevant from a clinical perspective, but when you then try to recreate that in a rigorous way, it becomes a challenge. It's very resource intensive to try to gather data from outside medical records if that is part of your mandate for the program. And then we have a high proportion of patients who are lost to follow-up in that process. So Genie is not alone in this program. There's many different institutions who are looking at the same sorts of problems. And this is just a sampling of them. And what we think is very interesting is that there's just different business models that are being proposed. There's different questions that are being asked. There's different reasons why you participate. And at Vanderbilt alone, we're participating in many of these consortiums. So we're participating in Genie, as I've said. We're participating in CancerLink. And the Tempus data collaborative and discussions with Tempus, they're all coming at your door, and it's very interesting as a medical center to think about, who you're planning to collaborate with and why and what your motivations are, because it's not without effort to participate in all of these programs. And the last thing I wanted to leave you with was that there are a lot of patient-driven initiatives that are also starting to come. So we've talked about institutions being data donors, but the next wave that's already starting to happen is really starting to see patients being the data donors. And can we, enable a crowdsourcing of patient experiences in that way? Count me in from the Broad Institute with their metastatic breast cancer project, their angiosarcoma project, and their recently launched metastatic prostate cancer project are similar projects. I'm going to leave with that, and I'll just thank all the Genie Consortium participants without which we would not be able to make all this progress. So thank you.